and findings and recommendations in terms of further research or clinical practice. The old sessions that we have here help us to understand what the science is and also provide an opportunity for the public to make any comment. The presenters this morning will have roughly 10 minutes apiece to make a presentation, and then at the end of each panel, we will have about 15 minutes that will allow the committee members to ask any questions that they might have of the panel. In general, we do not anticipate that there will be public questions to the panel, but if we have time and if somebody in the audience has a particular question, that will be perfectly appropriate. We need to remind everyone that any questions that you may hear, any discussion that you may hear on the part of the committee itself are the part of the process of us working through the literature and trying to understand the evidence, and you should not anticipate that this indicates any findings or conclusions that we have already reached. Understand that we have some people who are logged into this by audio. Wendy, I think you said we have maybe 20 or 22 people who are logged in. We don't necessarily know who these folks are, but the presenters should understand that your comments are being heard by more than just the people in this room. And am I correct? There is no provision for any questions or anything from the people that are logged on. So this is listen only. So with that, I would thank the panelists for taking the time to with us and to travel with us. I'm looking at where they've come from, and all of them have come from the Eastern time zone. A couple of them have avoided snow, so we know you're glad to be here. And so this panel will begin with Dr. Tom McAllister. And the ground rules that we've explained to them, they have 10 minutes. Morgan will be the timekeeper. No, somebody else is going to be. He will hold up warnings of 5 minutes and 1 minute. And when either Fred or I stand, you will substantially pass that 1 minute. All right, Dr. McAllister.
I'm tempted to do it just to see what that means, but I guess I better not.
Plug back there.
Yeah. The great gong will come out. Okay, so ordinarily trying to summarize this field in 10 minutes would be a daunting task, but it turns out that really there's not a whole lot of evidence to go on, so we're sort of okay. What I do is just sort of set the stage for why I think it's important. So from my perspective as somebody who's a clinician who's worked with folks with brain injury for 20-something years, one of the things I've always been impressed with is the variability in outcome. And this variability is, in a variety of domains. It's variability in risk for who gets a concussion. It's within individuals over time. So this person who gets a concussion in one phase of their life may seem to get be injured more easily as time goes on. So there's variability over time. There's variability in biomechanics. So we do studies with based accelerometers, and we can record people who are identified with concussion who have sustained a really hit, and then we can identify people who have been identified as a concussion who have had very small hits. Well, I wouldn't have hit, but nevertheless small in the overall scheme of things. So this has all raised the question about whether individual differences, in other words, the head that is injured, maybe plays a role in terms of both risk of concussion and outcome from concussion if injured. And, for you are going to be reviewing the literature in this and so forth, I wanted to put this slide in to emphasize that when we're talking about outcome, or even risk for that matter, it's a very complicated sort of multidimensional construct that we're talking about. And the effects of genetic differences in these areas are going to be very small. So it's easy for genetic effects to get overwhelmed by some of these other parameters which we know influence influence outcome. And therefore, in reviewing literature on this topic, it's worth asking yourself how well these other parameters have been controlled for in the papers, and whether or not you really can infer that these are genetic differences that we're talking about, or whether it might be just as easily explained by other factors. Again, 10 minutes, so I thought I would try and summarize all of genetics in 2 slides. But for those of you who haven't looked at genetics since high school biology, which hopefully is not many of you, but just wanted to make a couple of points, and that is that the genetic information obviously is contained in the DNA. The DNA can be present in a couple of different forms. It may be in the chromosomes that you're more used to seeing, at least from high school biology, but it can also be mute. Sweet. Okay. The DNA is packaged with proteins called histones, and it raises the question of how, so the DNA is there, genes are within there, how do you actually access the DNA, and how do genes get expressed? So it's not just the genotype that you have when you sustain your concussion, but it's also whether the genes that may come into play in terms of concussion risk or concussion outcome can actually be expressed. And that's important, is that the protein folding is relevant to how the protein does its task. And therefore, you can have a protein that is coded for example, in the cell that is active at a certain base rate at body temperature, and you can make a change in a single base pair, and now it is 4 times less active at body temperature. So it's important that just these single base pair changes can actually have what are called functional effects within the protein on the road. The thing is that it's not just genotype, as I said, is the gene being expressed. And this is a demonstration to show you that on the left, let's see, from you're looking at it, the left-hand panel is the DNA tightly packaged with the histone proteins and all folded up, and it's essentially impossible to get the genes there. And then on the right-hand panel is the DNA unfolded, and you access to the genes can get expressed and so forth. And what turns those switches on are the degree of methylation and acetylation of the histone proteins. So you can have the exact same genotype, 2 people, and one can have a difference in acetylation or methylation of the histones, and therefore that particular gene is being expressed more than in the other person. So it's not just the genotype, but it's the sort of epigenetic factors that you'll hear talked about as well. Okay, so if we then jump back to TBI and think about the context, the complex choreography of what happens in neurotrauma, we will not go into, but the bottom line here is that for every single step in the this choreography, there is a protein, and the genes which code for those proteins can have polymorphism polymorphisms, different forms, and therefore can be more or less active as a function of which genotype you have, and again, where that genotype is being expressed. Very, very complicated. It's not as simple as saying I have the good outcome gene or I have the bad outcome gene. That's the point. The other point is that, on the other hand, if you step back, there are certain broad categories of function in which genetic differences can, and the evidence suggests do, make a difference in terms of outcome. So genes which, for example, alter how much, what the response to neurotrauma is. So genes coding for the inflammatory proteins, the neurodegenerative proteins, the repair plasticity proteins, okay, all could be expected to play a role in not only risk, but degree of functional disability associated with an injury. Repair and plasticity, so the neurotrophins, for example, as an example, change in their activity might well influence how much the repair goes on, therefore how someone recovers. And then if you think about the genes available for before you even have your injury, so cognitive reserve, resilience, susceptibility to psychopathology, all of these have genes. So when you read literature, we're down to 1 minute, read literature looking at genetic effects, just bear this in mind, it's an enormously complicated array. That said, this is a summary of the literature that has taken this approach. I've organized it this way in terms of genes which alter response to neurotrauma, genes which alter repair and plasticity, genes which speak to cognitive capacity before you have your injury, so catecholaminergic genes, cholinergic system genes, and so forth. And all of these are papers that have been published with respect to influence on outcome after brain injury. The take-home message from this is that the proof of concept, I think, has been demonstrated, that individual differences, genetic differences amongst people who have a brain injury, including mild brain injury, do alter aspects of outcome after that injury, whether it's cognitive outcome or functional outcome or whatnot. Okay, what about sports concussions? And that was what we're supposed to talk about. We're running out of time. Here, very, very little data. If you think sports concussion is different than mild TBI or other types of TBI, so specifically populations or cohorts targeted to sports concussion, there were some earlier papers. Barry Jordan's paper looking at an N of 30 showed that there was an interaction in boxers between how often you got knocked out or were in serious bouts and ApoE4 status. So if you were hit a lot and you had the E4 allele, you had more neurological dysfunction. A study showed lower cognitive performance of old football players possessing the E4 allele. This is the allele associated with risk of Alzheimer's disease. And again, suggesting a potential interaction in that area. Terrell, and I'll finish, you haven't stood yet, but I'm almost, looked at a risk of concussion, and here I think you need to know that this was self-report, and for those of you who actually try and talk to people about how many times they've had a concussion, bear that in mind, that's sort of difficult, and actually found that there was no association between E4 status and risk of concussion by self-report in this cohort of college athletes. However, there is another gene in the promoter region of the ApoE gene, and they did find a risk of increased self-report of concussion associated with the T allele of the G219T allele. There was no association with tau polymorphism. This is interesting because tau is the protein associated with CE, or chronic traumatic encephalopathy. Very small cohort. So typically when you're looking at these kinds of studies, you're needing to look at much larger samples. And finally, Tierney's paper in 2010 showed a risk of concussion, again, self-report, again, N equals 195 or so. And here what they did, they looked at a variety of ApoE polymorphisms, so there's more than one in these genes. Typically there's lots of them. And what the headline was that 3 out of 4 who had all 3 minor alleles had concussions, but that's a small number, 3 out of 4. So the sample that actually had 3 of these abnormal genes had concussions, but I think it's difficult to draw any conclusions from that. So in summary, my view is that this field is in its infancy for sure. Of data out there, there is at least some promising and reasonably consistent signal both in TBI in general and a little bit in sports concussion in particular, that the ApoE4 or ApoE plays a role or is associated with parameters of outcome. What hasn't been looked at is additive or interactive effects at all. What hasn't been looked at are large sample sizes, so the largest sample that we have is 200 people. And there's even less evidence for in sports concussion, but there is at least a little bit. What's needed, large-scale collaborative studies, I think, using similar outcome measures, such as the common data elements that have been recently promulgated. Approaches that use not just a single candidate allele, but probably pathways geared at neuroinflammation, neurodegeneration, et cetera. If we could ever get a large enough sample size, GWAS studies, which are much more atheoretical but require samples in the size of thousands of people, would be ideal. And it's worth noting that there are several initiatives underway, not in sports concussion per se, but in brain injury as a whole that I've listed here that I think may help inform us on that. Epigenetic studies haven't even been thought out, really, and I would highlight the need to think about whether there's genetic variation associated with risk for chronic traumatic encephalopathy rather than assuming that everyone who has a brain injury gets CTE and so forth. And there are some references which provide a bit of an overview that will take you a little bit longer than minutes to get through. Thank you.
Our second presenter will be Dr. Inga Korte from Harvard Medical School.
Ladies and gentlemen, I